Thank you so much for your introduction, Javier. I am
Thank
Natalia, and
you.
I'm going to introduce my presentation, application of bioinformatic methods for the deconvolution of cell mixture to blood and immune cell types and comparison of generating gene signatures. The proportion of immune cell types in the blood can vary according to different factors like age, disease, or clinical interventions. For this reason, the analysis of cell heterogeneity provide us to know the changes produced in cellular diseases such as cancer or generated targeted immune response. In this way, personalized patient treatments. The analysis of cell composition is also related with RAS resistance. For example, specifically with immune checkpoints, both anti-PD-1 or PD-L1 and anti-CTLA4, when the levels of cytotoxic T lymphocytes are lower, whereas the infiltration of immunosuppressive cells are higher. In our work, we propose the convolution of bulk transcriptomic data to analyze the cell composition. It is a computational and mathematical tool able to decompose a complex cell mixture and identify cell types, calculating cell type proportions and or biomarkers expression from bulk transcriptomic profiles of heterogeneous samples. Considering N the number of genes and C the number of cell types, the convolution proposes a system of equation, one per any sample J, where its equation is the gene expression for individual samples. The gene expression is defined as the sum of the product of the gene signature expression in different cell types and the cell proportions for any sample J. The objectives of our work were perform a comparative analysis of the convolution methods, introduce and explain the convolution approach, assess the rate of 5 deconvolution methods in terms of immune cell proportions contained in the mixture, evaluate the precision using transcriptomic data with different complexity, select the best performing deconvolution method, and finally analyze the gene markers defined by the authors or identified by unsupervised methods. In our work, we use two types of deconvolution methods. In the first case, we use Cibersor, FARDIP, and ABIS as supervised methods. We solve partial deconvolution. In this case, we need the M or mixture matrix and S or signature matrix. And as a result, we will obtain the F or cell proportions matrix. In the second case, we use the conical lens set as unsupervised methods that solve the complete deconvolution. And in this case, these methods only require DM or mixture matrix. And we will obtain S or signature matrix and F or cell proportions matrix. In the first part of our study, we perform a comparative analysis using microarray signal expression data. This curve plot shows the correlation coefficients calculated between the real proportions obtained by flow cytometry and the proportion estimated by the CONICA, LINSET, Cibersor, and FARDIP. In general, all methods provide us high correlation coefficients, but LINSET didn't recognize T lymphocytes. This cell signature plot shows the proportions of real proportions in blue and the proportions estimated by the conical LINSET in red. We can see that the proportions inferred by the CONICA were distributed around an average value. Therefore, it not present a variability between different samples and cell types. We also observed that the similarity between distribution of cell types, the real cell types and the estimated cell types, decreased with low abundance. For example, the neutrophils usually were in the blood between 55 to 70%, but eosinophils were between 1 to 3%. And we can see that the distribution of between the distribution of real cell types and estimated cell types were more similar in the case of neutrophils than eosinophils. This bar mixture plot shows the proportions of different cell types estimated on the right and the real proportions on the left. It's important to note that the first sample of this data set was a tumor pure samples. Therefore, the immune cell types proportions should be zero. But Cibersort identify immune cell types in these pure tumor pure samples. But FARDIP can identify other cell types present in the sample, not defined in the signature matrix. In the second part of our study, we perform a comparative analysis using RNA-seq signal expression data. And as in the previous case, this curve plots shows the correlation coefficients between the real and estimated cell types for Cibersort, FARDIP, and ABIS. As an overview, all correlation coefficients were similar, but the values for Cibersort were a little lower than FARDIP and ABIS. In the last part of our investigation, we analyzed the cell markers through a signature matrix. In this part, we use the signature matrix provided by authors of Cibersort as a reference matrix. And we compare this signature with the cell markers inferred by Linset and deCONICA, both of them unsupervised methods. And we observed that all gene markers estimated by Linset were in the reference matrix, but only 67 to 500 genes inferred by deCONICA were in the signature matrix. Therefore, the Linset were more robust to identify immune cell markers. Sorry. As a final conclusions, all methods showed high correlations between estimated and real values. The proportions calculated by deCONICA were distributed around an average value. The methods recognized worse cell types that were not highly abundant in the samples. Cibersort was not effective for identifying tumor cells without defined marker genes in their signature matrix, overestimating other cell types. FARDIP was the best deconvolution method with high correlations, distribution of cell proportions similar to the real cells, and the powerful tool to identify other cell types not defined in the signatures. And finally, Linset was the best unsupervised method for identify cell signatures. As a future project, we propose investigate the composition of other cell types present in the tumor microenvironment, improve in tumor development like stromal cells or endothelial cells, study biomarkers that help to better characterize cell types, perform the convolution of myeloid cells. Now it's beginning to know next generation deconvolution, much more difficult to characterize and purely study so far. Explore the clinical outcome of cancer patients as a function of cellular subtypes or different oncology treatments. And finally, apply the convolution methods by implementing packages such as Immunodecom that integrate several of them, facilitating and accelerating the obtaining of results. And at the end, thank you so much for your attention. And if anyone have any question, I'm pleased to answer.
Thank you very much, Natalia. Any question in the audience? It's here.
Hi, Natalia. I appreciate your work, especially from the bioinformatic sides. I would like to ask you about, from your side, which are the features that may be contributed to downgrading the performance of Cibersort and methods like this? Which are the lines of research that you might pursue in the future to improve this resolution?
It's an interesting question, but I don't know. Do you mean that what is the best signature to perform the convolution?
Yes.
With Cibersort, for
how can you
example.
improve? Yes. How can you improve? Because you said that is an agreement between the estimation and the real numbers about and the real composition of the samples. But anyway, you still have difference to quantify.
For example, the signatures used by Cybersort is the most cell markers used. Moreover, these cell markers are used by other deconvolution by other authors of other deconvolutional methods, but for improve the signatures should be a deep investigation to, for example, with using single cell data
Of course.
for identifying the individual cell types.
Thank you.
Thank you. Any other question? Just a comment. I think Natalia showed it, but it's important, let's say, the result that we have, that Cybersort is probably one of the most common methods used for this deconvolution, cell deconvolution, but it's very much attached to blood cells, and it does not detect, for example, tumor cells or other types of cells. And then this is, from the point of view of the results that Natalia showed, I think is a relevant result, because other methods like FARDIP or LINSET are able to find other cells. And also in the cell mixture, the composition problem that Natalia revealed there is that the different proportion of the cells are very different. So sometimes, as she indicated, neutrophils are the main nuclear mononuclear cells in the blood, and other much in very small amount. So thank you, Natalia, again for the talk.
Thank you so much. Bye.